Welcome to The Explainer. I am so incredibly excited to have you here today. Look, if you've ever stood over a drooping, discolored houseplant or maybe a struggling vegetable garden with a sense of utter defeat, you are in exactly the right place. Today, we're going to radically change how you look at plant health. We are throwing away the panic, and instead, we're going to treat a dying plant with a forensic, highly methodical investigation. So, put on your detective hats, because we're going to crack the case of the dying plant together.
It's the ultimate question, isn't it? It's literally the question every single anxious plant owner faces when they see wilting leaves, bizarre spots, or stems that just don't look right. Your heart drops. You wonder if you watered it too much, or maybe too little, or if some invisible plague has just descended upon your garden. But rather than standing there feeling helpless, we need to completely shift our mindset.
Okay, let's dive right into this. The absolute core premise of our explainer today is that diagnosis is a forensic science. It is definitely not a guessing game. Far too often, gardeners just panic and resort to the guess and spray method, just grabbing whatever chemical is on the shelf and hoping for the best. Well, today, we are completely replacing that chaotic reaction with a systematic, evidence-based approach. To do that, we need a clear roadmap. We're going to cover 1. The botanical detective. 2. Gathering initial clues. 3. Abiotic versus biotic suspects. 4. Decoding signs and symptoms. 5. The disease triangle. And 6. Solving the case. Let's get into it.
Section 1. The Botanical Detective. Establishing the baseline.
I really love this quote from our sources. The diagnostic process is inherently forensic, requiring the collection of disparate data points to form a cohesive narrative of plant distress. This totally validates our whole detective framework. You've got to look at the big picture. You see, a classic mistake a lot of gardeners make is misidentifying normal genetic traits as a disease. For example, variegated thyme or certain hostesses naturally have these yellow-green patchy leaves. To an untrained eye, that looks exactly like chlorosis or some terrible viral infection. But if you know your baseline, you know it's just the plant's natural, healthy expression.
Section 2. Gathering initial clues. The investigative framework.
So the crucial point here is following a strict diagnostic protocol. Look at these 6 steps. Step 1, host identification, is, well, actually it's arguably the most important. There are hundreds of pests and diseases out there, but so many of them are incredibly host-specific. By properly identifying your specific plant variety right off the bat, you immediately eliminate a massive list of potential suspects. Then you describe the problem precisely. Is it on the new growth or the old growth? You analyze the spatial distribution and the temporal progression, meaning how fast is it spreading? Finally, you collect your physical evidence and do a contextual review of the environment, like checking recent weather patterns or looking into recent chemical use.
Section 3. Abiotic versus biotic suspects. Categorizing the culprits. In the plant world, the perpetrators fall into one of 2 major camps. Abiotic suspects are the non-living stressors. We're talking drought, terrible soil compaction, frost damage, or chemical drift. Biotic suspects, on the other hand, are the living organisms, the fungi, the bacteria, the viruses, and the hungry insects. As detectives, we absolutely must use the rule-out technique here. I really urge you to investigate the abiotic suspects first. Environmental stress is the most frequent cause of plant decline by far. Treating a frost-bitten plant with a chemical fungicide is not only useless, but it's literally a waste of your time and money.
Now notice how spatial and temporal clues act as a literal smoking gun. If the damage in your garden is uniform, like maybe it perfectly follows an irrigation line, or every single species of plant in a low-lying area is suddenly damaged all at once, that sudden, uniform damage is almost always an abiotic issue, like a sudden freeze or herbicide drift. But if the damage is scattered, sporadic, clustered, and it progresses slowly over time, spreading from one specific plant to its neighbor, that progressive timeline points directly to a biotic serial killer, like a spreading fungus or an insect infestation.
Section 4. Decoding signs and symptoms. Analyzing the evidence.
This is a distinction that trips up so many people, but it is absolutely critical for your investigation. A symptom is just a plant's physical response to stress. Wilting, that's a symptom. It tells you the plant is in distress, sure, but it doesn't tell you why. A sign, on the other hand, is the actual physical presence of the organism causing the problem. Confusing the victim's response, the symptom, with the actual perpetrator, the sign, can completely ruin an investigation from day one.
Let's train your eye a little bit. Fungi are the most common horticultural pathogens, and they leave incredibly distinct visual signs. Instead of just a generalized yellowing, you'll see definitive proof.
You might see dry lesions with clear borders, or these fascinating concentric target rings. On these infected leaves, you can clearly see the actual spore pustules, the fuzzy white mats of powdery mildew, or the bright, striking orange pustules of rust. Those aren't just symptoms, those are the physical structures of the fungi themselves.
And here is another undeniable sign. Finding an actual insect, like a caterpillar just happily munching away on a stem, is the absolute most direct proof of biotic interference you can possibly get. Even if the insect has already fled the scene, they almost always leave behind signs, molted skins, webbing, or frass, which, by the way, is just a very polite scientific word for insect poop. Seeing any of these signs confirms beyond a shadow of a doubt that you are dealing with a living pest.
But what happens when we don't have signs? Well, we have to rely really heavily on describing our symptoms accurately. Here's the professional vocabulary you're going to need. Instead of just saying, "My plant looks bad," you can observe if it has blight, a rapid death of tissues, or maybe chlorosis, that specific yellowing of normally green tissue. Is there a canker, a sunken, dead area on the stem? Or dieback, where the death starts at the very tips of the branches and progressively moves backwards? Using exactly the right words in step 2 of your investigation ensures you research the right solutions.
Section 5, the disease triangle, the master key.
Okay, this is absolutely huge. Most infectious plant diseases do not just happen by chance. For a biotic disease to actually take hold and cause severe damage, 3 specific mandatory components must align perfectly. First, you must have a susceptible host, a vulnerable plant. Second, you must have the pathogen actively present in the area. And third, you must have a favorable environment, like really high humidity or soggy soil, that actively supports the pathogen's growth.
And here is the brilliant realization that completely changes the game. If any one side of the triangle is missing, the disease cannot fully develop. Think about how empowering that is. You don't always have to wage this massive chemical war to eradicate a pathogen. If you can simply change the environment, maybe by pruning some branches for better airflow or just fixing your watering schedule, or if you simply pick a tougher, more naturally resistant plant variety right from the start, you break the triangle. And when the triangle breaks, the disease stops in its tracks.
Section 6. Solving the case. Integrated Pest Management.
Integrated Pest Management, or IPM, is how we officially prescribe the solution to our botanical mystery. It uses all the forensic data we've gathered to implement smart, highly targeted strategies. And notice the order here, because it's intentional. We prioritize non-chemical corrections first. We start with host selection, planting resistant varieties. We use cultural management, adjusting our spacing and watering to break that environmental side of the disease triangle. We can even lean into biological management by introducing beneficial bugs. Chemical management is always considered a last resort, used only when absolutely necessary and specifically targeted right at the verified suspect.
So, there you have it. We've cracked the code. You now have the full investigator's notebook. You know how to establish a baseline, spot the difference between an abiotic stressor and a biotic serial killer, and you know exactly how to break the disease triangle. So the next time you walk out into your garden and see a drooping, yellowing plant, I don't want you to feel panic. I want you to feel curious. Step into your garden with your new forensic toolkit and ask yourself, what are the clues really telling me? Thank you so much for joining me on this explainer, and happy sleuthing!